Hi, everyone. My name is Pang Kong Fox, and I am a parent to a child with a CACNA1A or CACNA1A related disorder. And I'm also the Science Engagement Director for the CACNA1A Foundation. This poster represents the Foundation's roadmap to developing specific treatments for the CACNA1A community. The CACNA1A Foundation was established in 2020 by parents of children impacted by CACNA1A related disorders. Our main mission is to develop specific treatments for the CACNA1A community through a collaborative network of various stakeholders, those impacted by this rare disease, their families, and researchers and clinicians. The CACNA1A gene encodes for the main subunit of the Cav2.1 calcium channel, which is globally expressed in the brain and important for neurotransmission or how neurons communicate. Information is sent along the neuron in the form of changes in the membrane potential. And once that signal reaches the end of a neuron, Cav 2.1 opens up to allow calcium ions inside, which induces the release of neurotransmitters or chemical messengers into the synapse and to the next neuron. Variants or permanent changes in CACNA1A have been linked to several neurological disorders over the last 25 plus years, but there are currently no CACNA1A specific treatments available for our community. These CACNA1A related disorders include ataxia and other movement disorders, seizures and epilepsies, hemiplegic migraine, global developmental delay, and intellectual and developmental disability, autism, and psychiatric illnesses such as schizophrenia. These CACNA1A related disorders are actually on the more common side of rare, with an estimated incidence rate of 1 out of 11,700 or 8 out of every 100,000 people. These CACNA1A related disorders are actually more on the common side of rare, with an estimated incidence rate of 1 out of 11,700 or 8 out of every 100,000 people. And as with many rare diseases, CACNA1A related disorders are underdiagnosed or misdiagnosed. If we take a look at ClinVar, there are over 550 pathogenic or likely pathogenic variants and over 1,400 variants of uncertain significance reported. So there's a lot of work to be done to get to treatments for our community. But the foundation has set a lofty goal to have at least one treatment in the clinical trial pipeline in the next 3 years. To achieve this goal, the foundation has spent the last 3 to 3.5 years working on two big initiatives: building a preclinical toolbox and starting clinical trial readiness programs like natural history studies. And the results are summarized in this figure, which is actually taken from our very first publication featured in Therapeutic Advances in Rare Disease. You can scan this QR code to access the paper, which has more details. This first box summarizes the CACNA1A Foundation's preclinical assets. The first chart shows the patient-derived iPSC lines that are currently available in the Foundation's biorepository. They represent a spectrum of variants, with both gain-of-function and loss-of-function missense variants and a frame shift that results in a protein truncation variant. These lines can be obtained by contacting us directly or combined brain. There are also a number of animal models available, many of which were actually established before the Foundation existed, and these are in academic labs or Jackson Laboratory. We also have several initiatives for understanding disease progression and burden, including a natural history study that is led by Dr. Wendy Chung at Boston Children's Hospital. We have just over 100 participants with a pathogenic or likely pathogenic CACNA1A variant. We also utilize the RareX data collection platform and have 120 participants enrolled in that. And we also take part in Citizens program to collect EMR or electronic medical records data. We've also launched our own quality of life survey, and a disease concept model study is in progress in collaboration with the Orphan Disease Center and Combined Brain. We've been working with Dennis Law and his team at UT Health Houston to develop a CACNA1A Data Portal. an open science tool that can be utilized by different stakeholders, including families, clinicians, genetic counselors, researchers, and industry. This work was funded by the Foundation and is the largest aggregation and curation of CACNA1A patient data. Data sources so far include published literature that spans over 2 decades and several clinical cohorts from Boston Children's Hospital of Philadelphia, and Cleveland Clinic. This also includes data from the CACNA1A Natural History Study and functional data from the work of Jen Pan at the Broad Institute. With this portal, you can search for specific CACNA1A variants, see the common HPO terms linked to the variant. You can compare variants across cohorts and other channelopathy genes as well. There are plans to expand data collection and also provide more granular phenotypic detail for each variant. You can scan the QR code to access the main portal. Please reach out to us at science@cacna1a.org to obtain access to the research tabs. It was through building this CACNA1A portal that we unofficially established our global CACNA1A Research Network. What began as a handful of researchers and clinicians who met monthly to help build the portal has now grown into a diverse network of over 60 researchers, clinicians, and representatives from regulatory agencies and industry that represent over 25 different institutions. We've held 3 in-person research roundtables, and we launched a series of CACNA1A working groups last year that meet monthly via Zoom. And we've achieved the following goals in just the last 9 months. Our working group, the Natural History Study and Biomarkers Working Group, designed and launched the CCARS, or the CACNA1A Clinical Assessments Research Study. The CACNA1A Variant Classification Group has developed a more comprehensive framework for variant classification. Our preclinical to clinical working group has developed a multi-lab drug testing pipeline that spans across diverse model systems. And our clinical care guidelines group has drafted the first clinical care guidelines for CACNA1A related ataxia, epilepsy, and hemiplegic migraine. The CACNA1A Clinical Assessments Research Study, or the CCARS, is a huge milestone for us. We launched this in July at our CACNA1A Family and Scientific Conference. This is a multi-institutional collaboration involving members of our CACNA1A Research Network and sponsored by the Foundation. And it is our effort to establish baseline measurements for our community using standardized outcome measures. This study also includes a comprehensive intake survey and the collection of additional clinical data, such as MRIs, EEGs, and videos of individuals in their native environments, all of which to help aid in treatment development for CACNA1A-related disorders. In summary, the CACNA1A Foundation's mission goes beyond advocacy and patient support. We have focused on establishing a patient-centric strategic research plan with a robust global research network, a comprehensive preclinical research portfolio, and emphasized the power of collaboration and data sharing to accelerate the path to clinical trials for our community. We are grateful to the Chan Zuckerberg Initiative for awarding us with a Rare as One grant. And we are thankful for our amazing CACNA1A community and research network for their help and support. And all authors on this poster declare that there are no conflicts of interest. Thanks for watching, and please reach out to us anytime at science@cacna1a.org or visit our website, cacna1a.org. You can scan the QR code shown here to get to the website.